So our next speaker is Dr. Michal Ben-Shachar from Bar-Ilan University. Michal completed her PhD at the Department of Psychology in Tel Aviv University, working with Professor Yosef Wojcinski, who is sitting with us today. She conducted her postdoc with Professor Brian Wandel at Stanford University, where she studied and continues to study the development of reading systems in children's brain. In 2008, Michal joined the Department of English, Literature and Linguistics and the Gonda Multidisciplinary Brain Research Center in Bar-Ilan. Michal's research at the Gonda Brain Center focuses on language and reading systems in the brain in healthy adults and children and in clinical population. And Michal will talk today about white matter pathways for language and reading studies in healthy adults, developmental and clinical population. And thank you for coming.
Let me know if you can hear me. Is that okay? Okay, so thank you for inviting me to this workshop. This is actually a great opportunity to provide students with state-of-the-art knowledge on diffusion imaging, and it's my pleasure to be here in the role of the application person. So I'm going to present several applications of diffusion imaging methods for the study of language and reading, mostly focusing on reading literature, not so much on the most recent studies that we've done, to save me something to talk about when I visit you on other matters that are more theoretical or academic. So my purpose here is going to be similar to a teaching purpose. I'm going to try to expose as many different applications, many different approaches to data analysis, and show how we can relate diffusion imaging measures to behavioral measures to learn something about the reading brain, the speaking brain. Hopefully we'll see how much we go. And I'll try not to go too fast, and if you have questions in the middle, please feel free to ask. So just to get started, to me, when we talk about higher and higher cognitive functions like language and reading, I think it's even more important to focus on white matter properties, because we're definitely not talking about the functionality of one part of the brain. We're talking about the whole system. If we're talking about a system, we might as well study not only the functionality of every single region, but also the connectivity between them. And this has been acknowledged as early as in Wernicke's and Lichtheim's days. Both acknowledge very clearly that we need to study connectivity. We need to model connectivity between brain areas in the goal of modeling language. Later on, Desjardins had similar ideas for reading. Another perspective that we've taken in the last couple of years is to talk about white matter impairments as the basis for various clinical situations or developmental deficits. Several learning disabilities have been related to reading, to white matter damage or to white matter deficiencies. Premature birth has been shown to be related to white matter impairments, and so it is of great importance to study the specific impairments and again relate them to behavior. And it's true even in the very recent models of language, for example, reading as well, that we're not anymore talking about a very simple model with two brain regions and a connection between them like in these days. We're talking about a more elaborate system, several systems actually, even when we're talking about language specifically. For example, this model from Hickok and Poppel shows us several different ways in which information that is transferred into the brain, let's say auditorily, can be processed, even if it's all in language information. It could be either processed along one pathway or another or both. And sometimes it reaches the pretty much similar areas, not to get into that debate too much, but it could reach similar areas using different pathways. And we're not anymore talking about a single path. We're not even talking about a single hemisphere. We have to include the right hemisphere as well and the connections with that hemisphere. Similarly, for reading, this is an image from Dahan's recent paper, not so recent anymore, Reading in the Brain, I think it's 2010, where he shows that the more modern vision of cortical networks for reading involves, again, dorsal systems as well as ventral systems, connectivity between many different brain areas. So we might as well study the properties of the connections, not only the properties of the brain areas. Having said that, I'm going to go straight into the study of the reading pathways using DTI. And this is a very well-cited first paper in which dyslexia has been examined using DTI. This was done by Torkel Klingberg in the very early days of diffusion imaging. He was a postdoc in John Gabrielli's lab at Stanford, and Torkel later continued to run many studies using DTI. In this first study, he recruited two groups of participants, adult dyslexics and control participants, very small groups, and he simply measured fractional anisotropy across their brains, and then voxel by voxel went ahead and compared between the groups. Okay, so we're talking about something very similar to standard voxel-based morphometry. You register all the brains to the same template, and then on the FA maps, you go voxel by voxel, and you ask, is there a significant difference between the dyslexics and the good readers? And you do that for every single voxel, and then you have, let's say, a t-value for a t-map for the entire brain. And using that simple method, Klingberg was able to find voxels that you can see here in the enlarged inset, in which he could show a difference between the two groups, where dyslexics had reduced FA compared to the controls. This is not all the regions that showed the difference. Actually, he found another area with differences in the right hemisphere, which is never shown visually and was therefore completely forgotten from the public memory. But this finding was even strengthened by finding not only a difference between the groups in this white matter area in fractional anisotropy, but also a correlation with behavior. So when Klingberg measured reading score on the y-axis versus anisotropy on the x-axis, you could see that there was a very nice correlation between the FA and the reading score. What sometimes people forget to say about this graph is that FA here is extracted not from just any region. It's extracted from the voxels that showed the highest correlation between FA and performance, and therefore we should be aware that there's very biased sampling here of the voxels, and that's probably the reason that we have such a nice, not only a group difference between the controls and the dyslexics, but also a nice correlation with behavior. But it's been till today our aim to show that you can correlate FA measures or diffusivity measures with behavior, and that behavior can go and become more and more specific. You may ask yourself, what is this reading score that they're using here? And it's a valid question. In the early days, these were very simple measures, and till this day, they didn't get to the point of cognitive sophistication that they can. In principle, today, I think we can take the behavioral part of this equation much further in terms of its sophistication, because we know a lot about behavioral measures. There's no reason to remain with the simplistic measures of behavior that come from our trying to stay with standardized testing, pretty much. So following this initial measurement by Klingberg, there have been, this is his initial finding, this is the center of mass of the area where he found the difference in FA, and there have been several different groups that immediately took this idea and went to try to replicate his finding in kids. And it's obvious, I hope, why you want to replicate such a difference between dyslexics and non-dyslexics. that was fine in adults in the developmental population, because when you see these differences in adults, they could be the result of lifelong dyslexia, less exposure to print. Many changes could have taken effect in the white matter of the brain in an adult, and therefore if you show this in a child, and taking advantage of the fact that we acquire reading quite late, you could sample these kids early on in reading development. If you find the same difference, it's a really strong demonstration that the difference is the cause and not the effect, okay? So the difference is white matter causes the difference in behavior, the disability in reading. So this was really nicely replicated by a group led by Christian Beaulieu in Canada, by Gail Deutsch in Stanford, working with Gabrielli and with Wendell at the time, by Niyogi and others with McCandless, and Nagy actually was working with Klingberg a few years later, found a difference in slightly different position, but notice the nice agreement between these four studies. I think this is, in DTI, this is considered tour de force. This is a great finding, okay? So replicability was achieved. One issue that we need to be worried about is that even though this finding is most commonly interpreted, as you can see in this image, this is a transparent brain trying to convince you that all the voxels that show the difference are actually along the arcuate fasciculus. When you look closer into the findings, you can see that with better tools than the ones that Klingberg had at these early days, you can see that the differences are rarely found in the arcuate fasciculus. They're usually found in the interface between the arcuate and the corona radiata, or straight on in the corona radiata. And these are areas that are well known to have many crossing fibers, and therefore the question was never solved at this point, and not to this day, whether we're talking about either of these two structures. Okay, I didn't remember this. Okay, so but the point is that several different interpretations have been offered for this finding. So it was very replicable, but the interpretation was difficult. Another thing I want to highlight at this point in time is that we're actually, so this is showing the same spheres, showing the differences between the groups, dyslexics and non-dyslexics, and now with respect to the to some fibers of the corpus callosum. Obviously, this is not a full picture of the corpus callosum, and similar to what Yaniv said earlier about the CST, the corpus callosum is an excellent example for how deterministic tractography sometimes fails to show some of the fibers. We all know that the callosum should be this beautiful butterfly connecting all the right hemisphere with all the left hemisphere pretty much, and what we see here is like a mohawk that connects the insides, the medial parts of the brain, but not so much the lateral sides, and that is because there are other pathways that prohibit these tracts from crossing from one lateral side to the other. So we need to keep in mind all the time that there are other fibers that we don't see with DTI, and we can say things about the fibers that we do see, but we shouldn't be interpreting the fibers that we do not see. I'll tell you more about this later on. Okay, so to move on a little bit, one question that comes to mind when we look at these findings is, where is the corpus callosum differences? And we expected to find corpus callosum differences because before DTI came to the sphere, people looked at the corpus callosum in just plain T1 and ran some analyses of the shape of the callosum, the size of the callosum, and it was a well-known fact about dyslexia, about the structural aspects of dyslexia, that the callosum is different in dyslexics, okay? When DTI first came into the sphere, this is where we would expect these findings to occur from earlier findings, and there were no findings at that point in time showing differences in the callosum. You might ask yourself why. The callosum is actually a place where we have very strong signal in diffusion imaging. It's an ideal place to test hypotheses. enjoy that area very much these days. And yet, when you look at this figure, it might help you see that there might be huge variability in the shapes of corpus callosum across individuals. So this is a figure, mosaic of 55 brains of kids that we've scanned in the year 2004. And each of them is shown in the mid-sagittal plane, and you could see that some of these corpus callosi have a very different shape compared to others. Some of them are completely arced, the other could be very flat. Sometimes the isthmus is very pronounced. Sometimes there is no isthmus or hardly any, like this one. So the shape varies a lot. When you take all these brains together and register them to the same template, you might find that the callosum is very blurry, or in other words, you might find that your power to find differences in the callosum between two groups is very low. And that is a result of registration issues, okay? Because when you have differences in volume and in size and in shape, you will have registration issues. And then the voxel-based methods are weakest, okay? So this is just a demonstration of calculating a power map. So we simulated a situation in which we took our data, we divided it into two subgroups of 30 kids in every subgroup, and we added, artificially added an effect. Okay, so we know that there's a significant difference between these two groups, but now after registration of both groups to the same brain, we can go voxel by voxel and calculate the power in every point in the brain, and you notice immediately that the callosum, shown here in the mid-sagittal plane, is very, very dark. The power is very, very low. There are two hotspots, one here and one here, where you're likely to find an effect if it's there. In the rest of the callosum, it's very difficult to find group differences and correlations with behavior, even if they exist in the data. Okay? So what should we do? Should we dump the callosum? It's such a nice part of the brain with diffusion, because anisotropy is very elevated in that area. All the fibers are coherent. We would really love to work on the callosum, and so I'm skipping the Whittleson example. And what we did was to track all the fibers that go through the callosum, and then rather than analyze each voxel of the callosum using voxel-based methods, we divided the callosum of every child in its native space into regions of interest. So we defined regions of interest using a method that was published a few years before this paper. These white matter regions of interest are drawn for every child separately according to gross anatomical markers. And then we can look at the callosum when it's now segmented into major segments. So, for example, all the green group is occipital fibers. The yellow group is posterior parietal, and so on. Okay? And the method we didn't invent, but we improved it slightly, because in the original method, I believe the entire frontal group was one segment, and we divided it. We continue to divide it a little further these days, because some of these segments are way too big still. So we can modify this this idea, but the method was very useful in now dividing the mid-sagittal part of the callosum, where the fibers are most consistent, all going in the same coherent direction, left to right, dividing it into specific segments. And we know how we found the segment, but now we focus only on the segmentation of the mid-sagittal plane. Okay? So this is for 4 different subjects. Each of them has its corpus callosum zoomed in on, and we can show how the segmentation I just showed for each subject separately divides the callosum differently. Okay, just looking at these enhanced images, you can see that a mathematical division, even if not voxel by voxel, a mathematical division according to the Whittleson idea, divide this into half, divide each half into thirds, and so on, is not going to work, because for some of these subjects, a group can be very small, or it could be larger, it could be shifted a little more forward or a little more posterior, And the idea is that across objects, the relative position of each segment is rather consistent. Okay? The size is not so much. This is when we did this for our entire sample. And I'm skipping a few details and jumping to the point where we looked for correlations with behavior in each of these segments. So we take the posterior segments where we expect to find any correlation with behavior based on predictions from Desjardins and others, based on predictions from Binder and others. And when we look at all these segments, the occipital segment, posterior parietal, superior parietal, or temporal fibers, we find that only in the temporal fibers there is a significant correlation between behavioral measurements of phonological awareness and the radial diffusivity within that segment. So the point, and I'm debating with myself whether I should explain what phonological awareness means, because it's sort of not extremely important to the point I'm trying to make. I'm trying to make the point that there could be an effect that you can measure as a correlation across a group that you wouldn't be able to measure in the average brain using voxel-based methods, because registration would just kill the power of that effect. And when you're analyzing it for every segment specifically that was defined for every child individually, you can reveal that effect that was transparent before. Okay? That was one segment. The second question that comes to mind when you're looking at this finding from Klingberg is, so how about the arcuate fasciculus? If we isolate the arcuate fasciculus separately in every child, can we find the effect, some sort of reading-related effect in that pathway as well? And so we did that, but we tried to do it in a more clever way. So to begin with, you have to consider what is the tract that we're talking about. And when you're looking at these voxels and you're looking at this image, it was very clear to Klingberg and others that they would like us to think this finding relates to the superior longitudinal fasciculus, which is a major connection that connects actually all three major lobes. It connects between the frontal lobe and the parietal lobe. There's a branch, another branch that connects between the frontal lobe and the temporal lobe, and a third branch that I'm not even going to touch on that connects the most posterior two regions. And so when we thought we should really test this prediction from Klingberg that the arcuate is going to be impaired, we thought, let's just isolate this one, the frontotemporal part of the superior longitudinal fasciculus, and ask the question selectively on that part, because that's the prediction that we're going to be talking about language connections between the inferior frontal areas and the posterior temporal areas. Okay, so the question is the arcuate fasciculus, you could call it the long segment according to Catani, is it mediating reading signals or reading-related signals? And this was work done by Jason Yateman. He took the data that we had from these 50-something kids that we scanned. At that point in time, we already scanned them a few more times. And Jason found the arcuate fasciculus in every one of these kids, as you can see here for 3 kids. And this is just a heat map of where these tracts meet the cortex. And I was surprised by your heat map that was so condensed. When you take all 53 kids, it's rather a mess in terms of where they end in the temporal lobe. Was that one for only one subject? Okay. So for one subject, it looks very focused. And when you take all kids together, you could see a lot of variability in terms of the temporal endpoints of these pathways. And you can see that in terms of the frontal endpoints, they tend to connect pretty much the precentral gyrus and the most posterior part of the pars interocularis with various parts of the temporal lobe. Okay? we looked at correlations between FA and phonological awareness in this tract. When you average across the entire tract, you find modest correlations. They go negatively, which is a pain because it's very hard to explain why less diffusivity, why less anisotropy will predict better performance. But I'll get back to that later. What's more important is that the correlation is not so great. There's huge variability here in where the points are spread. And one possible reason for that is that when you look at the anisotropy along the tract, you see huge variability. This is along the nodes are different points along the tract, okay? So in some points along the tract of the arcuate in one subject, you can find very high FA going all the way up to sometimes even 0.7. In other parts of the tract, the same tract, in a single individual, you find sometimes a drop to almost 0.3. This was mentioned here earlier, that within a tract, you can find huge variability. And if you do this for one child, this is one source of variability. When you look at the other kids, you can see that sometimes the profile is similar, but they're not registered to each other, okay? So that's another, if you do this for 50 kids, you see that that this is a miss. When you average across the whole thing, it's actually a great surprise that anything comes out from the average number of FA for every individual and behavior. And even if you look at the profile of how FA varies along the tract, it is not a very good representative of individual variability at any point. Sorry, so what we did here, if you want to understand exactly why FA drops, well, FA drops because at certain point in the tract, as Katani has pointed out, the two tracts diverge. One of them goes parietally, the other goes temporally, and at that point, crossing fibers just drop FA altogether. So that's one source of variability, and I can tell you more about other sources of variability, including myelination, coherence, and whatever. So we did something that we thought would help. We took these profiles and we used the point in which the tracts diverge. And that is also, by the way, the point of maximum curvature of the tract. We used that point in order to align between the profiles. And then we didn't look at that point to look for correlations because that point FA is very low, and it is not a good representative of anything about the tract. It's mostly a representative of how much crossing there is. So we looked elsewhere in areas where FA was high, around here, and in those areas, FA is higher. There is likelihood that we're actually measuring properties of the same tracts of white matter. Okay, so extracting FA from here, we could find a stronger correlation, still negative, stronger correlation between FA and phonemic awareness that we measured in the same set of kids. And that finding, you know, when you're solving a crossword puzzle and you have the first letter and it matches another word, then you're pretty sure that your guess was correct. That finding, the fact that you're doing something in the right direction and the effect improves or gets stronger, that gave us the feeling that we might be in the right direction. We still had to face this issue of why is this correlation going down, and till this day we sometimes find negative correlations, sometimes find positive correlations, and it's a big issue in the literature of how to interpret those. But what we did here, luckily, was to scan the same group of kids several times. We actually scanned this, and during my postdoc I scanned these kids 4 different times along the years. They came every year for a scan or 2 and behavioral measures. And so we could assess their reading development as well as their brain development. And when you're looking at the changes, not the FA at a given point in time, but the change in FA over time in correlation with measurements of how their reading changed, you can find a positive correlation, which is what you would expect, not only in the arcuate fasciculus, but also in another pathway in the ILF that connects occipital areas with more anterior temporal areas. And together, the ILF, this is taking together the contributions from the arcuate fasciculus and the ILF, they explain a nice portion of the variance in the reading score. Okay, this is the predicted reading score based on the ILF and Arcuate fasciculus anisotropy, and this is the reading score that resulted. All right. So you might ask yourself, well, we only sampled the temporal tract, and I'm glancing over a lot of the detail because we did sample other tracts to make sure that this is not a difference or a correlation across the whole brain. It's selective. This is just one specific question you might ask. Can we show that different segments of the superior longitudinal fasciculus are related to different properties in behaviors? Basically, we're trying to do some sort of a dissociation here, functional dissociation, between very adjacent pathways, trying to relate them to different functional properties. Okay, and I already showed you that the temporal segment is related to phonological properties of the kids. I can tell you that when we looked at the frontoparietal segment, there was no significant correlation. Neither when we looked at the right hemisphere. The connections in the right hemisphere did not correlate with phonology as well. And when we looked at math, mental math, in the same group of kids, only one of the years, we added some mental tests, some mental math tests, we can find a correlation in the frontoparietal segment with measurements of math, specifically when kids were required to estimate rather than calculate. We could find a correlation with FA from a segment of the frontoparietal connection. We only went specifically to that pathway because in the Hans work, many times we could see that mathematics activated, or numerical cognition activated, regions in the inferior frontal and inferior parietal areas that are commonly connected by this pathway. And so there was a hypothesis that led us to look at this pathway, but while we were there, we also looked at the long segment, the Arcuate fasciculus, and we didn't find a correlation for math with the long segment. So this is now a functional dissociation that I'm trying to convince you of between the two different segments of the SLF. I'm saying maybe here because in a few recent papers that I've seen, this segment is also sometimes related to reading, and so I have to figure out what is wrong with my dissociation. For me, this was very logical because the connections between Broca's area and temporal areas should have a lot to do with phonological calculations, whereas if you're talking about the connections to inferior parietal, less so, maybe. So I want to just summarize up till now. What we have shown is that you could relate phonological awareness, a behavioral measure of reading-related skills, not reading, but reading-related skills in kids, with diffusivity properties in some segments of the corpus callosum. What we're doing here, let me say very generally, is using tractography. Amir, this goes to your question earlier on. We're using tractography in order to identify the regions of interest. And even beyond that, within the tract, we're using an analysis that segments the tract so that we can identify an ROI within the tract for every individual. And having done that, we extract the numbers, the FA or radial diffusivity, whatever, and we try to correlate that with behavior. And I think it is important to remember when you're looking at tractography results that I think about it almost like I think about parametric maps in fMRI. The map is not the data. Okay, this is not what the paper is telling you. You need to look at the numbers that are extracted from these regions. Hopefully, if they were defined individual subjects, then this is not all about registration. It's about the individual properties. And I have an example of how looking at whether you find a tract or you don't find a tract as the data, I think this is wrong. This is an image from Daniela Perani and Angela Frederici and others, a paper that was published in PNAS. What they did was amazing. They scanned newborns that were born in Italy. They, anyway, as Perani tells it, when you go to the mothers and you ask them, can I scan the brain of your, these are healthy kids. Apparently in Italy, the moms are just saying, okay, you can scan the baby. So they went ahead and they scanned the baby in Italy, and they found that the pathways, they got, of course, Helsinki permission and everything they needed to do, but still, it's pretty amazing that this can actually happen with healthy babies. They scanned the newborns and they tracked fibers, and they found the inferior fronto-occipital fasciculus, both sides, and they found the arcuate fasciculus, but as they say, it was a shorter arcuate fasciculus, and it didn't quite reach where it usually ends in our healthy adults. And these are adults that were scanned in Germany in a different scanner. And using those methods, they could find two different arcuate segments, one that ends in the pars opercularis, the other that ends in the precentral gyrus. And so the main argument of the paper is that the newborns are missing the blue branch. And the first point that I want to make sure that I pass along is that not finding a pathway is never an indication that it's not there, because it's very likely that we're failing to find a pathway, especially in newborns, because there's not enough myelination, because our methods are limited. They use deterministic tractography. There is nothing fancy about the method. There is nothing that shows that you could use this data to show that the fibers are not there. So this means we didn't find virtual fibers, but it doesn't mean that the actual axons are not there. And the second point that I want to highlight is that our level of angular resolution is such that when the fibers get close to the cortex, they could go slightly to each direction and hit one gyrus or the next gyrus. And it's very rare that we can talk with great precision and confidence about these choices that the fibers make. And so even in adults, I'm not completely sure that there's evidence that there are two separate segments of the arcuate fasciculus, but rather it's possible that some of the tracts take one choice and others take another choice, and maybe these are the same axons, and this is the property of the tracking algorithm and not of the data itself. I think we need to be very careful about the points that are at the border, at the limits of our resolution, at the limits of the methods that we're using. And it's important to know the limits of the strength of the methods before you go ahead and overinterpret data. Okay, last part of my talk is I want to give you like very like the highlights of a recent study that that we've done. And I tried to have a methods point for everything. So the methods point here is that sometimes diffusion tensor imaging can actually detect new tracts, and Catani has done that for a while. This is a paper from Deschatin and Catani, where they claim that they discovered a new tract that they call the Frontal Aslant tract. Sometimes these tracts have their precursors in dissections, sometimes not. But they find it both in atlas data from monkeys provided from Schmaman and Pandya, but also using diffusion imaging in the human adult brain. And they don't only describe the pathway, but so they go through a lot of details in describing the pathway, and that's very useful for us when we try to find it again. This pathway, the frontal aslant tract, connects between the pars opercularis or triangularis, so Broca's area, broadly speaking, a little posterior to that sometimes, and the SMA, the supplementary motor area. The other thing that they discovered about this tract, this is a paper from Brain 2003 by Catani and others, is that the frontal aslant tract was impaired in a version of primary progressive aphasia, specifically in patients that had a hard time talking, okay, non-fluent PPA. So primary progressive aphasia is not caused by stroke, it's caused by degradation of the brain in aging, but it's not natural aging. It's definitely a degenerative disease that causes non-fluency. So these patients produce language very slowly and with great difficulty. And when they look at that group of patients and look at the average properties of the L-aslant, they found that they have reduced FA. The other groups of patients did not show that difference. So they deduced that the L-aslant tract likely has to do something with producing language. Vered, who's sitting somewhere here, wanted to test whether, if this is the prediction, can we show differences between adults who stutter and controls in that specific tract. And very briefly, I'm going to glance through a lot of the methods of how we did that because it's late. What we did is to track fibers in the whole brain, which we usually do. We did the same with the callosum and only then restrict to the callosum. Yaniv said earlier that if you track fibers from the whole brain, your computer will crash. I can tell you this used to happen in 2006. But these days, we can definitely track fibers from the whole brain. If you're using a Linux box, it will hold, even if you're subsampling each voxel. So we are tracking fibers from the whole brain. Then we define regions of interest that are defined on a template once. And then we work these regions back to every individual according to a back transform between the individual and the template. And finally, we use these ROIs in order to segment that subject, the specific subject tracking results, in order to find the arcuate fasciculus, the frontal aslant tract, my apology. Okay? So this is the frontal aslant tract in one individual in the left hemisphere. This is in 8 individuals in both the left and the right. The ones on the top are stuttering adults. The ones on the bottom are control subjects. I didn't tell you much about how we selected these participants, but in the final analysis, we had something like 15 adults who stutter and 19 controls. And we then extract the mean diffusivity and the mean anisotropy from the entire tract. And already at this point, you can see that in the left, fat means frontal aslant tract. In the left fat and in the right fat, you can see group differences between adults who stutter in red and controls in blue that were significant in the mean diffusivity of these subjects. No difference was found at the mean level in anisotropy. And similarly, other pathways that Vered looked at did not show similar differences. Next thing that we did is when you look at the profile of mean diffusivity along the entire aslant tract, you can find a significant area where the groups diverge. And when Vered extracted the mean diffusivity of only that area that showed the significant group difference, she could show correlation in the stuttering group between the mean diffusivity and their speech rate. So those that speak faster have reduced mean diffusivity, which in a perfect world will say enlarged anisotropy, but not in this group. In this group, diffusivity and anisotropy were not that beautifully correlated. So the difference here, the correlation and the difference here were with mean diffusivity, and I was happy to hear Aviv saying earlier that mean diffusivity is his favorite measure, or Yaniv said that, and we supported it, and we are in favor of it too these days. At least the direction makes perfect sense, because higher mean diffusivity means that there is higher mean diffusivity means that there's less barriers to diffusion, which could mean more myelation. It could mean various things. I hope I didn't get the directions correct. I got the directions correctly. So to summarize, I just wanted to give you a few examples of how we use diffusion imaging in order to study language pathways, reading pathways, not only the most familiar ones, but also some that have been recently supported. I think it's important not to discover too many new tracts and study them before you do the groundwork and make sure, or somebody else does the groundwork for you, and shows that there's good reason to believe that this pathway really exists and that there's some emerging ideas about what its functionality, and then you can find ways to converge, to use converging operations in order to deduce what kind of functionalities are related to that pathway. Something that was on the slide, on a previous summary slide, and I didn't mention, is that I think it's important for us not to make a mistake and think that if we find a correlation with behavior, it means that this pathway is doing X. Okay? A pathway can do many different things, similarly to a brain region, but definitely when we're talking about a pathway, we're talking about several different functionalities that could be context-related. We shouldn't base our deduction solely on one correlation, but we should look at the same pathways, many different tools, and get to a generalization about which functions it's related to in specific contexts. So I know this would make our stories much more complicated, but I do think that it would be a mistake to say right now the L-aslant is for speech rate. I think that's not the correct interpretation of the data. All right, and I just want to thank my students. Many of them are here today. Ofer, Amir, and Ruti Ezrati from Tel Aviv University were our clinical accomplices, helping us to define what is stuttering and what is not. I worked as a postdoc in Brian Wendell's lab for about 5 years, and this is when we did the longitudinal reading study that I told you about. And the scanning that I presented from Vered's work was done in Ichilov. Thank you. Happy to take questions. Yes?
Yeah, my question is, did you, by any chance, look at the relationship between the differences you find in dyslexics in white matter and the differences that exist in gray matter? So the retracts that you highlighted throughout these studies, as you know, is very close to the planum temporale.
Right.
The question is, did you relate these things?
Okay, so I only, okay, let me say this. I measured fMRI signals in the same kids, but the question was focused not on regions that do phonology. Our questions were focused on the visual word form area, and therefore we used very visual types of manipulations. So these manipulations hardly activate the temporal areas. However, in a group of maybe 8 kids, I ran rhyming and various localizers like that, but that was not enough. When we looked, we tried to relate those because at that point we realized that the temporal segment is the most interesting one. So relating it with their functional signals in 8 kids, that didn't work. It's not enough. What we did, anatomically, exactly. So anatomically, we again wanted to ask your question. It makes sense that if you find in the temporal group a relation to phonology, it should be one specific part of the temporal lobe, not the entire, you would hope. And so what we tested was as follows. We tracked fibers from Wernicke's planum temporalis, sort of posterior part of the superior temporal between the two hemispheres using probabilistic methods. And we did the same for another region in the temporal lobe that was not there. It was empty because we had other findings in MT related to motion perception, whatever. And indeed, what you find is that the pathways from superior temporal to superior temporal, posterior third of the superior temporal in both sides, are the ones that pass through the same area we are talking about. And I believe also they increase the correlation that you find with behavior, whereas the other one is not the same area of the mid-sagittal callosum where we saw the fibers going, the empty, like motion processing area. Okay, so structurally there was a match. Unfortunately, this is not published because it was a postdoc that left before she got to publish it, but I know that the results were there. Yeah, I think there is a lot to do in order to connect DTI more broadly. Connecting DTI to functional signals is something that is done not very often. In several papers where it was done, there is, at least for functional connectivity analysis, it wasn't a good match. And so I think we still have to figure out how to combine fMRI and DTI, not only to use fMRI as the regions of interest to restrict DTI, but actually to combine the signals into one analysis. And it's not trivial. We tried a few things, and it's a challenge. Okay, maybe we need to use not FA, but more specific quantitative measures like Excalibur, because Yaniv's example of how they combined Excalibur with ERP that measures the speed of transmission through the callosum, that's a nice example of combining. Other questions? No? You're tired?